So now we continue our today's lecture that is all about the proteins. Um, First of all, I'm sharing the screen to you.
Okay.
Is the open board visible to you?
Yes.
Okay, whiteboard is visible, right?
Yes.
Okay, fine. So in the start of the lecture, first of all, I'm taking a recap like quiz. That is a very simple type quiz. So I have like some questions relevant to the last lecture, which we are taking. So we have three questions. The first one is the what is the difference between the essential and the non-essential amino acids? Like, just tell me like any two amino acids, or just tell me the concept like what are the essential as well as non-essential amino acids?
Okay. So the essential amino acids, basically, they cannot be synthesized in our body. So they must be like we have to consume them in order to obtain them. So I think some examples were leucine and then lysine.
Yes,
And then
exactly.
the non-essential amino acids are ones that can be synthesized from our body and they don't need to be obtained from food. Like there was alanine and then glutamine
Yes,
for
exactly.
the example.
Yes, exactly. Very good. Now tell me the classifications of amino acids.
So the first classification is whether they're essential or non-essential based on whether our body can synthesize them or not. The other is based on their chemical composition, which were non-polar. Non-polar ones are hydrophobic. Polar, which were hydrophilic. Whether they're acidic, and then whether they're basic.
Yes,
And then,
exactly. Continue, please.
oh yeah, and then for what is a peptide bond is the next one. So a peptide bond is a covalent bond, and it's formed between the amino group and the carboxyl group. So it's the one amino acid forms a bond with the carboxyl group, and it releases water, and it
Exactly.
forms proteins by linking amino acids.
Yes, exactly. And when multiple amino acids combine by a peptide bond, they form a
A polypeptide bond.
yes, exactly, polypeptide bond. So now we are moving towards the first slide of the today's lecture. That is all about the classification of protein structure. We classified the protein structure into two major categories. The first one is the globular proteins, and the other one is the fibrous proteins. So first one, the globular proteins are actually it has a globe-like structure. That is why we call them the globular proteins. So the structure is the globular proteins are the round structure, and they have a round structure because the hydrophobic part of the protein fold inward, while the hydrophilic part means the water loving part, and the polar part of the proteins become arranged outward and around the external surface. This means that on the outer side we have the water loving and the polar protein part, while on the inner side the hydrophobic part, this the hydrophobic part is present on the inner side. Because of this these proteins are globular proteins are the round like structure. Actually why they are round from the outer surface, because the outer surface is like polar and it easily makes bond with the highly electronegative atoms like the oxygen, nitrogen, sulfur and phosphorus. And because of this, because these structures are present on the outside, that is why the structure is a little bit round. And the another structural property of the globular protein is globular proteins are the water soluble. Means that as only the hydrophilic component of the proteins are on the outer surface, we already said that on the outer surface of the globular proteins the hydrophilic part is present, means the polar part is present. And because of this hydrophilic component on the outer side, globular proteins are the water soluble component. Like any compound which have the highly electronegative atoms like nitrogen, sulfur, phosphorus and oxygen, when it comes towards this round surface, this external surface, so the polar part of the polar surface of this globular protein easily attract these all and make a bond with all of them. So that is why we call them, they are the soluble, like globular proteins are the water soluble, and they are soluble in water. If we are discussing the functions of the globular protein, so first of all, due to their round shape and the soluble nature, globular proteins play a vital role, a wide variety of vital metabolic roles in the human body. The first one is the enzymes. That is very basic concept about the proteins, that all the enzymes are the globular proteins. That is very important. Actually, all the enzymes are the globular proteins, as their round shape and surfaces can be altered approximately to fit their target site with with high specificity. This is the picture of the like enzyme. Like these all are the proteins. Like this is the protein and the enzyme. This is the enzyme, and it has a specific this actually represents that an active site completely fit into the enzyme. But the main concept we are taken from here is that uh, an enzyme have a specific globular like round shape, and due to this round shape any active site can properly fit into it. Like this is the active site of the enzyme. So any like this is the substrate which react with the enzyme. So the substrate like easily adjust in this position of the enzyme. So because enzymes have the globular structure, and it altered it can be altered approximately to fit the target sites with a high specificity. Another like justification is that like enzymes की round surface होती है, वो अपने आप को like उस active site के accordingly altered कर लेते हैं अपनी shape को. Like जिस type का active site आपको मिलेगा, तो उससे होगा कि like जिस type active site enzyme को मिलता है, enzyme अपने आप को उसके accordingly alter कर लेते हैं. That is the basic like essential and sequence of the enzymes in the proteins. So the example is the amylase, pepsin, and the lipase. These all are the protein enzymes which break down the starch, protein, and the fat respectively in our body. The second function of the globular protein is the transport proteins. Transport proteins means due to their soluble nature, as we know that globular proteins are soluble in water, and due to their soluble nature, globular proteins functions well as a transport protein, as they can cross all membranes. Like they can move across all the membranes. As an example is the like hemoglobin, which transports the oxygen. And we have another example, like this is the lipid barrier. We already discussed in the lecture of like the carbohydrates. This lipid barrier have a protein channels. These are the like this one is the protein channel. This is the protein channel. So it can transport the molecules in and out of the like this lipid barrier in and out of the cell. So they also work as a transport medium. It can transport like molecules in and out of the cell and across the cell membranes. So the globular proteins have like two functions. The first one is the first one is the first one is the enzymes. Like all the globular enzymes are the globular proteins. And the second one is the transport protein. Like protein enzyme have sorry proteins have the like globular proteins have the ability to pass out the molecules in and out of the cell membrane. So this is all about the globular protein. The next classification of the protein structure is about the fibrous proteins. Fibrous proteins are simple. They are made up of the repeated amino acids sequences. Like we have this one amino acid chain. Like the amino acid combines with another bipeptide bond and they form a long chain through a sequence. So they are made up of the repeated amino acid sequence that form a long polypeptide chain. And these chains twist together to form the fibrous protein. Like this is the one chain of the polypeptide. Like this is the one chain of the proteins, and this one is the another chain of the protein. So these two chains become twist towards like each other, and they form a fibrous protein. Like in the form of like this, and in the also in the form of like DNA. These two chains of the protein, they twist toward each other and they form a fibrous protein. These are the long chains polypeptides, and these are the long chain proteins. The another one is about that proteins, fibrous proteins are the water insoluble. Means that this means that on the outer surface we have the on the outer surface of the fibrous protein we have the hydrophobic part, hydrophobic part. That is why they are water hating and they are water insoluble. As hydrophobic part of the polypeptide chains are not folded away from the external environment, fibrous proteins are not soluble in water. Like the hydrophobic part are not folded inward and they are present on the outer surface. That is why the fibrous proteins are the water insoluble. And when we are moving towards the functions of the fibrous proteins, so the first function is, as we know that the fibrous proteins are the stable as well as the insoluble structures, so they are not suitable to function as a metabolic protein. That is very important for metabolism. And for metabolic proteins the globular proteins are here, but in case of the structures the fibrous proteins work. Rather they act well as a structural protein which support and protect the tissues. These fibrous protein protects our hairs, protect our nails, like these all things. So the example includes the keratin, which provides a structure to the hair and the nails, and collagen, a type of the connective tissue that is present in the body. So fibrous proteins are the long chains, and these are the long polypeptide chains that twist around each other. The second important thing is that fibrous proteins are water insoluble. Although the globular proteins are the water soluble, and the function is like fibrous proteins work as a structural protein, while as the globular protein work as a metabolic protein. And the examples here, the examples are the keratin and the collagen. Means as a like keratin which provides a structure to the nail and hair, and the collagen which provide the connective tissues in the body. This is the these are the example pictures of the fibrous proteins, like that polypeptides twist around each other. And the other one is the example of the globular proteins. So this is all about the classification of the protein structure into the globular and the fibrous proteins. Have you any query?
No questions so far.
Okay. So the next topic is about the levels of the protein structure. In the level of protein structure, we have like four major levels of the proteins. The first one is the primary structure. The second one is the secondary structure of protein. Then the tertiary, and the last one is the quaternary structures of protein. First of all, we again memorize the structure of the amino acids. Like in the structure of the amino acid, we have a central carbon atom that is connected with the three permanent groups. The first one is the amino group, the second one is the hydrogen group, and the third one is the carboxylic group. And we have a one R group, that is the variable group. So we call them as the variable group. And these three groups, like the amino group, the hydrogen and the carboxylic group, and the central carbon atom, we call them as the backbone of the amino acid. This means that why we call them backbone, because this major portion is present in the all the classes of the amino acid, whether they are polar or non-polar, or whether they're essential as well as non-essential amino acids, or either they are charged or uncharged amino acid. But this portion, this like permanent groups in the central carbon atom is present in all the amino acids. That is why we call them the backbone of the amino acids. And the R is the variable group, like it changes in every amino acid. So we have like four structural levels of the proteins: primary, secondary, tertiary, and the quaternary. First of all, we will discuss the primary structure of protein. So in the primary structure of protein, the sequence of amino acids in a polypeptide chain held together by the peptide bonds only. This means that this is the structure of the a sequence of amino acid in a polypeptide held together by peptide. Yes, in the structure of the like in the primary structure of protein, like this is the protein, and these proteins are combined through the peptide bond. So this is the complete a polypeptide chain. The concept of the primary structure is that in the primary structure the protein the amino acids are combined by only the peptide bonds, and this is the like one protein. And if we have the same another chain of the protein, that is the another chain of protein, so these two chains of proteins are only held together or only combined by a bond that is called as the peptide bond. Like if they both are combined with each other, or they both held together with each other, they are only held by the peptide bond. No any other bond is present, like the hydrogen bond or the disulfide bond. No other bond is present in it. That is why we call it the primary structure. Like the sequence of the amino acid in the polypeptide chain held together by the peptide bond only. This means that when polypeptide chain is only made from the peptide bonds, no other bonding is involved, and it mostly have a straight chain and mostly have a linear structure. So this is the primary structure of protein, in which only peptide bond is present. The next one is all about the secondary structure of proteins. So when the polypeptide chain is linked by the peptide bond as well as the hydrogen bond, this means that in the secondary structure of the protein, we have like two type of bondings are present in the structure of the protein. The first one is the peptide bond, as it already present in the primary structure of protein. The first one, and the second one is the hydrogen bonding. So these two are the bonding bonds that are present in the secondary structure of protein. The proteins do not exist in just simple chain of polypeptides, like they don't have just a like a peptide bond. These polypeptide chains usually fold due to the interaction between the amine and the carboxylic group of the peptide link. Actually, what happened here is, this is the secondary structure of the protein. In the secondary structure of the protein, we have we show that we assume that like this is one amino acid. This is one amino acid, and this one is the another like second amino acid. And we said that like from first amino acid, the NH group, like here NH is present, like NH means the amine group is present, NH2 amine group is present, and here the carboxylic like C double bond O group is present. These two, like the nitrogen and the carbon, they make a bond with each other through the hydrogen bonding. We know that in hydrogen bonding, hydrogen is bonded with either nitrogen, sulfur, phosphorus, or oxygen. Means hydrogen is bonded with the highly electronegative atoms. That is why we call them the hydrogen bonding, because hydrogen is bonded with all of these. So in this structure, nitrogen is like bonded with any nitrogen is not, like the carbon is bonded with the hydrogen of this hydrogen of this amine. Means that, like for example, a hydrogen is present here and the carbon is present, so this make a bond with each other. So majorly we said that the amine group of one amino acid and the carboxylic group of the other amino acid combines, and they form a bond that is called the hydrogen bond. So these polypeptide chains usually fold due to this bonding, this hydrogen bonding. The both amino acids are folded towards inward. That is why we have like a coil like structure in the secondary structure of proteins. Due to interaction between the amine and the carboxylic group of the peptide link, the structure refers to the shape in which a long peptide chain can exist. They are found to exist in two different types of structures. Like they form two different types of structures. First of all, we have to clear that in the secondary structure of protein we have like two types of bond. The first one is the peptide bond, and the another one is the hydrogen bond. And now, due to this hydrogen bonding, this polypeptide chain becomes folded towards inward, and they changes their shape. So they changes their shape in the like two forms. The first one is the we call them the alpha helix, and the second one we call them the beta pledged, like beta pledged. The second one is the beta plated sheet. So the first one is the alpha helix and the beta pledge. Like due to this hydrogen bonding, the whole polypeptide chain like moves towards inward, and they like convert itself into two different forms. Like some of them are the alpha helix and some are the beta plate sheets. Now we are discussing both of them, but this structure arises due to the regular folding of the backbone of the polypeptide chain due to like hydrogen bonding between the carboxylic group and the amine group of the peptide bond. The next slide is about the alpha helix and the beta plate sheet. Actually, a polypeptide can form a coil-like shape that is called as the alpha helix. when a polypeptide chain can form a coil-like shape, that is called as the alpha helix, and when it folded like a fan-like shape, it called as the beta pledge. Like this is the structure of the alpha helix. It fold itself into a coil-like shape. This is a coil-like shape. It folded itself into a coil-like shape. But in the case of the beta pledge sheet, it folded itself into a fan. Like this is like a fan, like handy fan. It's like a fan. So they convert itself into a fan-like shape. That is called that is why we call them the beta pledge sheet. So further we will study these like alpha helix is one of the most common way in which a polypeptide informs all the possible hydrogen bond, but that is important, like by twisting into a right-handed screw with the amino group of each amino acid residue hydrogen bond to the carboxylic group of the adjacent turn of the helix by twisting into a right-handed screw. That is why that we call them the alpha helix. And the polypeptide chains twisted into a right-handed screw. In the beta plate sheets, in this arrangement, the polypeptide chains are stretched out beside one another and then bonded by intermolecular hydrogen bond. Like they are stretched out beside one another and they are bonded by the hydrogen bonds. In this structure, all the peptide chains are stretched out to nearly the maximum extension they can and then laid side by side, which is held together by the intermolecular hydrogen bonds. And the structure resembles the pledge folds of Draffry, and therefore it's called as the beta pledge. Like they form the like fan like structure, and they are stretched out nearly to the maximum extension they can. That is why we see the structure like this. Like maximum जितनी stretch हो सकी थी तो ये sheet जो है stretch हो रही है. So this is the beta pledge sheet and this is the alpha helix. So first of all, we will study about the primary structure of protein, in which only peptide bond is present between the chains of the polypeptide and no other bonding is present. But in case of the secondary structure, we have two types of bond which are present. The first one is the peptide bond and the other one is the hydrogen bond. But due to this hydrogen bonding between the carboxylic group and the amine group, two different types of structures are formed. The first one is the alpha helix and the other one is the beta pledge sheets. So the alpha helix is like a coil shaped structure, is like a coil like shape structure, that is called as alpha helix, and the beta pleated sheet is like a fan like shape. That is why we call them the beta pleated sheet. So this is all about the secondary structure of protein. Now we are moving towards the tertiary structure of protein. Have you any query about the primary and the secondary structure of proteins?
No questions so far.
Okay, so third one is all about the tertiary structure of the proteins. So this structure arises from further folding of the secondary structure of protein. Like this structure arises when you are further folding the secondary structure. Like number of possible bonds are present in the tertiary structure of protein. It has the like peptide bond. It has a peptide bond as well as the peptide bond as well as the hydrogen bond. Both the bondings are present. But now in the tertiary structure, the other bondings are also present, like the electrostatic forces. The third one, the electrostatic forces. Fourth one is the disulfide linkages. The fourth one and the fifth one is the van der Waals forces. These bondings are also present in the tertiary structure of the protein. So the tertiary structure of the protein resembles overall the folding of the polypeptide chain. Further folding of the secondary structure, it gives rise to two major molecular shapes like fibrous and the globular protein. Fibrous and the globular proteins are actually the classification of the tertiary structure of proteins. And the main forces which stabilize the tertiary structure of protein are the hydrogen bond. Another one is the disulfide linkages, and the third one is the van der Waals and the electrostatic forces of attraction. So this is the like complete structure and overall structure of the tertiary protein. But this is the internal structure of the tertiary protein. In the internal structure, we see that like we have multiple types of bonds. Like now we understand that, for example, here a protein is present. here a protein is present. So maybe this is the like non-polar amino acid, like this is the non-polar amino acid, and this one also is the non-polar amino acid. And because of this they are combining by the aliphatic compound. Like here the R group is the aliphatic compound, and because of this they create a hydrophobic interactions. And here a hydrogen bonding is present, means that it maybe it has the amino acid which have the functional group like the hydroxyl functional group and any other like which have oxygen bond. So because of this these two amino acids combined by the bond that is called as the hydrogen bond. For example, we take the example of wait, yes, in the last slide the pictures. Yes, these are the pictures. For example, a glycine, which have the functional group that is the hydrogen, and another one is the serine, that is the functional group OH. So maybe these two amino acids combine, and obviously they are combining by the bond that is called as the hydrogen bonding. So here, like the hydrogen bonding is present in it, means that like for example, glycine and serine are present. Because of this, they form the hydrogen bond, and here a disulfide bridge is present. Why the disulfide bridge is present? Maybe here an amino acid is present which have the disulfide functional group, like where the disulfide functional group is present. Like this is the structure of the amino acid which contains the disulfide functional group like acid. That is the example of the cysteine. Cysteine has the self-hydryl functional group. Self-hydryl means disulfide. In disulfide we have like two sulfur, or maybe like the disulfide bridges are also formed because of the cysteine or the one having the self-hydryl functional group. So these two amino acids are combined because they have the first the sulfur is present in their functional group, and it also have the ionic bonding. Like how ionic bonding is present between the two amino acids, because these two amino acids are the like polar charged amino acids, like the one having the positive charge while the another one which have the negative charge. And because of these positive and negative charge an attraction is developed in it, and because of this an ionic bond is formed. So these all types of bonding, like the ionic bonding, the hydrogen bonding, disulfide bridges, hydrophobic interactions, and the peptide bonds. So because all of this, the structure of protein is found that we call them the tertiary structure of protein. Means that because of all these bondings, the secondary structure of protein is further folding, and because of this complex folding, we call them the tertiary structure of protein. And the last structure is about the quaternary structure of protein. In the quaternary structure of protein, it is about like it is the aggregation of two or more polypeptide chain into a single functional unit. Like we have, you assume that we have multiple polypeptide chain. Like this one is the chain of a polypeptide. Like this one is the complex tertiary structure. This one is also the complex tertiary structure, and this one is also the complex tertiary structure of the protein. When all these tertiary structures of protein combines, and they all combines, they make a single functional unit. Like this one is the like this is one tertiary structure of the protein, is the another tertiary structure is the another, and this one is the another. So these four tertiary structures of protein, when they all combine with each other, they form a single functional unit that is called as the quaternary structure of proteins. So like in quaternary structure of protein, we have like same bondings are present, like the hydrogen bonding, peptide bonds, disulfide bridges, ionic bond, van der Waals forces, and the last one is the hydrogen bond. So these all are the same bonding are also present in the quaternary structure of proteins, but the difference is that like number of the tertiary structures of the proteins combine into a single unit, and they will form the quaternary structure of protein. So this is all about the levels of the protein organization. The first one is the primary, another one is the secondary, the third one is the tertiary, and the fourth one is the quaternary. So everything is clear to you?
Yes.
Okay. If I am I have a question from you, like how many levels of the protein structures?
There's 4 levels.
Yes, four levels, and what are those?
The primary, secondary, And then there's a tertiary and quaternary
Yes,
structures.
exactly. And in which structure of protein, in which level of protein, like maximum number of the bondings are present?
In quaternary,
Yes, quaternary, and also in the
uh, also in the
tertiary.
tertiary, yeah,
Yes,
tertiary.
because like the number of bondings are same in the quaternary and the tertiary, but in the quaternary, like all the primary secretaries, all the secondary structures of proteins are combined into a single functional unit. But we cannot like count the bondings. We said that like how many like bondings are present in the form of like the first one is the hydrogen bonding, the second one is the peptide bond, third one is the like dash peptide, the fourth one is the hydrogen bonding in the same like the fourth one is the ionic bonding. So they both have like four or five different types of bonding. So in that sense I'm talking about you that like which one have the maximum number of bonds. So in the quaternary and in the tertiary they both have like same types of bondings. All right.
Yeah.
Okay. The last point is about the functions of the proteins. Proteins have so many functions in our body, like in the bodies of the humans, in animals, in plants, and like in others. So they have a lot of functions. Proteins are very diverse. They are highly diverse macromolecules. Even they are like diverse from all the other classes of the macromolecules, like from the carbohydrates, lipids, and the nucleic acids. This specific class, like protein, is highly diverse class of the macromolecule. They perform the essential functions in the body, and they can be classified based on their roles as follow. The first function of the protein is about it catalyzes the chemical reaction, like enzymes and the proteins that speed up the biochemical reactions. For example, when we eat any type of food, like we are eating the fruits, so from like our throat to our small intestines, at different specific positions, number of different enzymes are present that break down our food into the smaller pieces and like convert to the smaller pieces and like pass out to the lower portions of the body, like from throat to the stomach, and stomach to the small and the large intestine. So actually the enzymes and the protein that speed up this biochemical reactions. Obviously when we have a like very large component of the food, like this is the complete component of the food, we have like a fruit and apple. So obviously this is this cause a very like slow amount of reaction, but when it breaks down into the small pieces, definitely it definitely like it readily absorbed. So enzymes are the proteins that break down the break down the chemicals, and it convert into the smallest part, and it speed up the biochemical reaction. For example, digestive enzyme like amylase and lipase, they break down the food. So the enzymes are the proteins that help in the biological reactions. For example, this is the graph that shows the enzyme action. When we have a protein, we have like any chemical, and when it passes through the like body, and when no enzyme is present, like it has no enzyme, this causes a very long and a very slow reaction, that when it move towards the upward, like we are supplying the energy to this reaction, and then it slows down very slowly, and now the energy is released. So this has a very high activation energy without the enzyme. But when the enzymes are present, the reactant like glucose plus oxygen, they will speed up the reaction is like speed up. They readily show their reaction, like the reaction is like when we supply the energy, and in a very highly speed they releases their energy and convert into the product. So enzyme have the function like they speed up the biochemical reaction. So the enzyme is the like proteins. The second function of the protein is about it provides the structural support. Like protein fibers provide the strength and the support in various structures. For example, they provide the strength in the bones, in the tendons, in skin, in hair, in the nails, in the claws, in the beaks, and in the bones. In all these regions of the body, they provide the strength and the support. Like collagen is the connective tissue, and keratin the in hair and the nails. Like keratin is the protein that is present in the hairs and the nails, and they like strengthen our hairs and the nails, and the collagen is present in the like connective tissues of our muscles. So proteins have another function, like they provide the strength and support. Like this is the example of the nails. So like the nails have the keratin. The first function of the keratin is like they provide these strength. Is like some little bit not a clear picture. In the first function of the keratin is it provides the strength. Like for weak damaged nails, it provides the strength. The second function is about the hydrate. For the dry and the brittle nails, it hydrate the nails. And the third function of the keratin is it provides the growth. Like for thin and the peeling nails, it grows those nails. And the last function is the maintenance. Like it maintains the nails growth. Like for nails in need of the nourishment, it maintains the nourishment. So so the proteins have the function, like they provide the structural support to the different areas and the different regions of the and the different components of the body. The third major function of the protein is like it act as a transporting medium. Like it transports the substance in the body, and that is like the proteins help move the molecules inside and outside of cells. Proteins are helping the substances to move like inside and out of the cell. For example, we are taking example of the lipid bilayer, and like this is the structure of the lipid bilayer. Like we have a protein channels, and through these protein channels, these are the extracellular fluids and these are the intracellular fluids. So this means that from these protein channels, these proteins are passing out from these channels, and it not like pass all of the cells. It selectively pass some cells, like some allow some cells to pass from these channels. So proteins also work as a transporting substance in the body. For example, hemoglobin is the protein that transports the oxygen in the blood. And another information is that membrane protein regulate the entry and the exit of the substances in the cell. The fourth function of the protein is about it enables the organisms to move. Like the actin and the myosin, which both are the contractile proteins, which are responsible for the muscle contraction. Like I'm giving to you the example from the daily life. First of all, actin and myosin are the two essential contractile proteins that play the crucial role in the muscles contraction and the movement in the animals. Like when actin move towards inward and then myosin move towards the outward, like they both moving there in the opposite directions. So the best example from our daily life is like when you walk or run, your legs muscles contract and relax due to the sliding movement of the actin and the myosin filaments, and your calf and the thigh muscles repeatedly shorten and lengthen and enabling the forward movements. Another example is like blinking and smiling. That when the small muscles like when small muscles do contraction, their contraction is controlled by the actin and the myosin, which allows your eyelids to blink and the facial muscles to form the expressions. So these are the proteins, like these are the contractile proteins, and when they contract, they perform the function. Like the example of the blinking and the smiling, and the example of the running and the walking. The another one is the regulating cellular processes. Like proteins have the function that they regulate the processes, cellular processes. Like some proteins act as the hormones to send the signals between the cells. Like the best example is about the insulin, which is the insulin is the hormone which tell you about the sugar level in your body. If the sugar level is like much lower, like the insulin regulates the blood sugar level in your body. So hormones are also the proteins. They act as, like these are the proteins which like working in the way that they send the signals between the cells in your body. The last one is about the it provides the defense against the disease. Like this is very important. Proteins are providing the defense. They have a defense system against the diseases. Like antibodies are the proteins that helps the immune system fight infections, and it protects against the bacteria and the viruses. So these all are the examples of these all are the functions of the proteins. Like they catalyze the chemical reaction in the form of the enzymes. They provide the structure support in the form of the collagen and the keratin. They act as the transporting substance in the body in the form of like hemoglobin transport the oxygen in the body. They are like working as the contractile, like muscle contraction, and they are working as a and the contractile proteins responsible for the muscle contraction. For example, the when we are running and when we are walking. And another function is about like it regulates the cellular processes. Like insulin is the best example. Like the proteins act as the hormones, and when any part of your body feel low, or when any part of your body like feel the a little bit low, then these hormones send the signals to those specific cells, and those cells like those proteins overcome their efficiency. And the last point is about the it provides the defense system against the diseases. Means antibodies are the proteins that help the immune system to fight the infections, as well as they work against the bacteria and the viruses. So it's all about the functions of the proteins. The next point is about the learning checkpoints. Are you understand about the functions of the proteins?
Yeah.
Okay. So the last point is about the structure, about the learning checkpoint. So the first question is like explain using at least four functional examples. Okay, first of all, I'm telling you the question, like question number 23. So summarize the four levels of the organization in the protein structure. Maya, I have a question that summarize the four levels of
Oh,
organization in
yeah.
the protein structure.
Okay, so the four levels would be the primary, secondary, tertiary, and quaternary levels of organization. The most bonds are present within the not the primary, the tertiary and quaternary, and which they which means that they have the most amount of bonds present between the protein. They also, in terms of structure, it was that the primary has a singular bond, and secondary would have two bonds. And I forgot some of the features of the structure. That part I'll need to review.
Okay. Uh, like question number one, what is the significance of an R group? That is the variable group in the amino acid. So what is the significance of an R group?
So the R group basically represents the difference between each of the types of proteins, because the functional group is attached to the R group, which could be a different group of the number of carbons, which gives it the different properties.
Yes, exactly. And explain using at least four functional examples, like why proteins represent a diverse type of macromolecule.
Okay, so for this, there's the diverse so the different functional so we have the enzyme proteins, which
Yes,
will speed up chemical reactions. We
exactly.
have structural proteins, which provide support and strength to cells and tissues, like collagen.
Yes,
Oh, I forgot for the one above, it could be like amylase. The transport proteins, which help substances move across cell membranes, which would be like the hemoglobin. And then hormonal proteins, which
yes,
help with like growth and metabolism, like insulin.
exactly. So now the answer which I written here is for like we are just revised these all. The first question is about explain using at least the four functional examples why proteins represent a diverse type of the macromolecule. So proteins are the diverse macromolecule because they perform the various functions. The first function is the enzymatic proteins. They catalyzes the biochemical reaction. For example, the amylase. The second function is the structural proteins. They provide the support and the strength, like collagen in the connective tissues. The third one is the transport proteins. They carry the molecules like from into and out of the cell, like the hemoglobin transports the oxygen. And the last one is the hormonal proteins. It regulates the psychological processes like the insulin controls the blood sugar. The second question is about the describe like using a labeled sketch the basic structure of an amino acid. So in amino acid, the structure is like that. We have a central carbon atom that is bound with the four different groups. The three of them are the permanent groups, like the amino group, the carboxylic group, and the hydrogen group. But the fourth one is the variable group. This is also called as the variant group, and this is the R group where the functional group is attached. The next question is about what is the significance of an R group, like the variable and the variant group. So the variant and the variable R group determines the chemical properties and the functions of an amino acid, influencing the protein structure and the function. So the next question is about like explain why proteins are more structurally and functionally diverse than the carbohydrates and the lipids. As we know that the proteins are the more diverse than carbohydrates and the lipids because they have 20 different amino acids, allowing the complex structure. They fold into the specific 3D shapes, enabling the diverse functions, and they form the enzymes, receptors, structural components like carbohydrates and lipids, which primarily serve as the energy and the storage roles. So that is why they are structurally and functionally diverse from the carbohydrates and the lipids. The next question is about this how summarize the four level of the organization in the protein structure. So the four level of the protein structure is about they have we have four levels of protein structure. The first one is the primary, second one is the secondary, the third one is the tertiary, and the last one is the quaternary. Like in the primary structure, sequence of the amino acid in the polypeptide chain is judged by the peptide bonds. In the secondary, local foldings occur into an alpha helix in the beta sheets due to the hydrogen bonding. In the tertiary structure of protein, the three-dimensional structure is formed by the interaction between the R groups due to different number of bondings, like the hydrogen bond, the ionic bonding, the disulfide bridges, and due to the ionic, like the hydrogen bonds. And the last one is about the quaternary. So multiple polypeptide genes assembling into a functional protein like the hemoglobin. So this is the like the four levels of the protein organizations in a protein structure. And the last but not the least, the last question is about explain how the three-dimensional structure of a protein can be changed in the absence of a gene mutation and why this can be harmful to an organism. That is a little bit conceptual question. Like explain how the three-dimensional structure of a protein that can be changed and why this can be harmful to an organism. This means that when any structure of protein changes due to any condition, like how this that specific protein is harmful for that specific organism. So a protein's three-dimensional structure can be changed due to the denaturation. This is the another concept that is about the denaturation. This means that when like denaturation is the concept that when any protein changes its shape due to any cause, like caused by the heat or changes in the pH or the chemicals, it leading to the loss of the function. This means that when any protein changes its structure due to any cause, like by heat, pH, or the chemicals, it leads towards the loss of the function, complete function changes, the complete function losses of that specific protein. And this can be harmful as enzymes in the structural proteins lost their biological activity, affecting cellular function and examples. These are the two examples from our daily life that when the protein denatures, like what happened with the organism. So the first example is about the high fever and the enzyme denaturation. When the body temperature rises about 40 degrees centigrade, during a high fever, enzymes in the body begin to denature because of this high temperature and because of like a high heat pressure. This like the enzymes are denatured in the body, losing their shape and the function. This slows down the metabolic reaction, leading to fatigue, dizziness, and sometimes organ damage. And another example of this denaturation is about the sunburn and the skin damage. So excessive ultraviolet radiations from the sun can denature the structural proteins like the collagen, and the elastic in the skin, leading to the sunburn, premature aging, and the skin peeling. So because of the degeneration of protein, and whenever a protein changes its shape due to any of the reason, so very harmful reactions are occur on the body of an organism. So this is the last question from the learning checkpoint, and this is where our topic like the protein is completed. So if you have any query, please ask from me.
I know so far I have no questions on it.
Okay, and is these notes are like helpful for you?
Oh, yes, they're very helpful.
Okay, so this means that we are covering all the topics like introduction of protein, classification of the proteins into globular and the fibrous proteins, the functions of the proteins. The other one is the amino acids, like the monomers of the protein, like the polar and the non-polar amino acids, as well as the essential as well as and the non-essential amino acids. What are the polypeptides, levels of the protein organization, and the learning checkpoints? We discussed all of them and we understand all of them. If you have any query, like when you are reading it from the book, so just text me on the Skype, and if you are like asking in the like during the lecture, so just ask for me. For this is for about today's, and in the next lecture we are covering the next macromolecule and the last macromolecule, which is about the nucleic acid. All right.
Yes. Also,
So
after that, we have like one more topic in school before we do our quiz, which would be like biochemical reactions. But after we do like those two topics, would it be possible to do like a mock test on what we've learned so far, just like before Thursday? Because I have my quiz on everything we've learned up till biochemical reactions on Thursday.
yes, inshallah, I'll arrange a like mock test for you so
Okay,
that
thank you.
your preparation becomes more well.
Okay, thank you.
But, okay, inshallah. So tell me the date, like on what day I'm arranging the mock test.
So it would be best. I have classes on Wednesdays, so Wednesday class.
No, we'll take on Tuesday to this week
Okay.
because you have a test. So Monday, Tuesday, and Wednesday she has classes with you, and Thursday too.
So I think Wednesday would be best because Monday and Tuesday we'll do the two other units.
Yes, yes,
So
okay.
Wednesday
Make
would.
sure on Wednesday we are arranging the mock test.
Yeah.
Okay, inshallah. So thank you and Allah Hafiz.
Thank you, Allah.